All right, so we're going to talk about meiosis today. We've already talked about mitosis, which is just a copy machine, right? You have one cell, the DNA makes a copy of itself during the S phase of interphase, and then it undergoes undergoes the stages of mitosis, and then we have two cells at the end. So if I have a cell with 48 chromosomes, say like in the bone of a person, and they break their bone and they need to repair the bone, those cells will undergo mitosis to repair and recover that injury. So then we have new cells that are exactly like the starting cell. So mitosis is a copy machine. 48 chromosomes in a body cell. At the end of mitosis, we have how many cells? We have one cell with 48 chromosomes at the beginning. How many do we have after mitosis is complete? Two. And how many chromosomes are in each cell? How many? 48. Exact copy machine, right? So this is where we see throughout the body. Mitosis is occurring throughout the body, except for places like we said, the neurons and the muscle cells and fat cells. They do not divide, even though they shrink and grow depending on what's going on in the body. For as far as fat and muscle, they don't undergo mitosis. Neurons don't either because we have just a set number. That's why we got to protect the neurons we have, right? We know that alcohol and marijuana kills brain cells, so that's why it's important that we respect the number of brain cells that we are given, right? Some people don't do that as well. So meiosis then is a similar process of dividing cells, but the goal is different. In meiosis, we do not get identical copies. We get diversity at the end of meiosis. And instead of 2 cells, we get 4 cells at the end of meiosis. So that's a big difference, is the number of cells we get at the end, and they're not exact copies. Third major difference is we have 48 chromosomes at the start of meiosis, just like in mitosis, but at the end of meiosis, we have half the number. So we would have, what would it be? 46, I'm not 48, 46 chromosomes. Oh my gosh, sorry. 46 chromosomes. At the end, we have 23. So what do we call those cells that only have one set of a chromosome? Haploid cells. And the symbol we use for haploid is N. Okay, so at the end of mitosis, we have 2 diploid cells, and at the end of meiosis, we have 4 haploid cells. So the goal of meiosis is not for repair or growth or replacement. It's to produce haploid gametes. So when we talk about gametes, we're referring to sperm and egg. So at the end of meiosis, we start out with a cell that has 46. At the start, we have a cell with 46 chromosomes. It undergoes the 2 major phases of meiosis, meiosis 1 and meiosis 2, and at the end, we have 4 sperm cells, each with 23 chromosomes. Now, in the egg, it's a little different. In the egg, we have 46 chromosomes in the beginning, and at the end of meiosis 1 and 2, we have 4 cells, but one is going to be a functional egg or ovum, and the rest are called polar bodies. They're small, non-functional cells that we don't need. So if you think about it, which do we need more of? Do we need more sperm or more egg at any one period of conception? One egg means one baby, right? We don't have lots and lots of babies, right? So producing fewer eggs in meiosis is not a big deal because a woman only produces so many eggs in her lifetime that are capable of fertilization. But males need lots of sperm with every meiotic division because it's a long way to travel and not every sperm can make the trip. So there's a lot more sperm than there are eggs that are produced at the end of meiosis. But regardless, there are 4 cells at the end of meiosis, whether you're making egg or sperm. But 3 of those cells, like I said, in the female are non-functional. We call them polar bodies. And we'll get more into that. So the goal is to produce haploid gametes. And those gametes, when they're haploid, 23 and 23, they come together at conception. What we say conception is when a zygote is formed. That's when the sperm nucleus enters the egg and those nuclei fuse. So now you have 23 chromosomes from dad and 23 chromosomes from mom fusing together to give 46 chromosomes for baby, right? So that's diversity. That zygote is a mixture of the chromosomes of the mother and the father. So the zygote is diploid because you have 23 and 23 coming together. Okay, so when you look at our chromosomes then, this is what it looks like in a human cell. We have 23 different chromosomes. You can see this is a pair. The 23rd pair are the sex chromosomes. So the 23rd pair can be two X's or it could be an X and a Y. If it's two X's, like in this diagram here, that means this individual is a female. If it's an X and a Y, then that individual is a male. So we can look at a karyotype of DNA in a fetus and determine if there's any chromosomal problems to make sure by looking and making sure that there's one of every chromosome and they all look about the same. But again, this chromosome number 1 has different traits on it, and that's why it has different color bandings. There's different genes that code for different traits on each of these different chromosomes. And there's a lot of traits on chromosome number 1, wouldn't you agree? It's a huge chromosome. So there's lots of DNA, lots of genetic information about this individual on chromosome number 1. Look at chromosome number 21. Very tiny, right? Probably not a lot of information there. So when there's chromosomal problems, like babies that inherit an extra chromosome number 21, because maybe the spindle broke off in meiosis, they can end up with 3 chromosome number 21s. They didn't separate in anaphase 2, and then that person has what we call Down syndrome. Maybe you've heard of that. You probably have heard of that and seen individuals with Down syndrome. So because that chromosome is so tiny, that those individuals that have an extra chromosome 21 can survive fetal development and make it to birth and live and have a job and family and friends, right? Because there's very few traits carried on chromosome number 21. It's such a small chromosome. But there are enough trait differences that you can definitely tell someone who has Down syndrome. And as a nurse in the hospital setting, we know that people with Down syndrome often have heart dysfunction, heart abnormalities. That's part of this extra chromosome number 21. And they also have cognitive delays, varying levels of that. And they have facial features that are very specific. And what else? They're prone to cancer. So there's some side effects of that extra chromosome that we see. So where did one of these chromosomes of number 1 came from mom, the other one came from dad. So together they're expressed depending on, we talked about homozygous recessive, right? So if chromosome number 1 carried a recessive allele, the one from mom and the one from dad was also recessive, that person is homozygous recessive for that trait on chromosome number 1, right? But if chromosome number 1 carried a trait that was dominant from one parent and recessive from the other, what do we call the individual then? If it's like big R, little r. What is the term for one of each? This would have been on your lab exam yesterday. Heterozygous, right, heterozygous. So when we talk about traits and how they're inherited, we're talking about alleles that are carried on these chromosomes that we inherit from our parents. So we're really a combination of our parents that way. So we call these chromosomes, the 2 chromosome number 1s, we call them homologous chromosomes because they carry similar traits, similar information, but it's not exactly the same, right? Because we're talking about widow's peak on whatever chromosome that's on, dad could have a widow's peak, which would be big W, dominant, and mom may not have a widow's peak, which would be little w, right? What is the individual then with those 2 alleles combined? If dad was big W, mom was little w, what is baby going to be? Because remember, the sperm has half the chromosome. So if the sperm was little w and the egg was big W, what was baby? Big W, little w, which means heterozygous. Right, and are they going to have a widow's peak? Yeah, if they carry that big W, that dominates over the recessive, so then they would have a widow's peak. So looking at chromosomes then, each pair that we see here are called homologous chromosomes. Homologs, homo means same. So they're chromosomes that carry the same information. The centromere is located in the same spot, and we call them together chromosome number 6 in our karyotype, which is a map of our chromosomes as we see here. These are homologous chromosomes. But this chromosome here and this chromosome here are not homologous, right? Because they carry different traits and they're different chromosome numbers. Okay, so they have the same length. One came from mom, one came from dad, centromeres in the same spot. And when we stain them, they have a similar pattern. So we already talked about a typical chromosome is just single-stranded DNA, coiled up, condensed DNA like a spring. And then when it replicates in interphase, it forms this X-like structure, right? And each of these is considered a sister chromatid. So this might be from dad, this chromosome, and this might be from mom, this chromosome. So that's why we call these homologous. Same type of information, but one from dad, one from mom. And when they're in the X form, it just means it's been replicated in interphase. All right, so the different variations on homologous chromosomes we call alleles. So we talked about if they're the same allele, same letter, big T, little t, those are homozygous. Or if it's big T, little t, the chromosome from the mother had a recessive allele, and the chromosome from the father had a dominant allele, then the individual is heterozygous. So now we're going to just talk about some major processes. When you look at meiosis, it's two different state or phases or events. There's meiosis 1 and meiosis 2. A lot of stuff happens in meiosis 1 that I'm going to hold you accountable for on the test. Like prophase 1, something special happens. Metaphase 1, something special happens. So you're going to have to pay attention to those details. And then we get to meiosis 2, it's kind of like mitosis. It's very similar to mitosis. So if you understood mitosis, you're going to understand meiosis 2, because it's just a repetition of cell division. So we're going to watch an animation of meiosis, because I think that's the best way to understand it. But the key thing is we start out with a cell that has diploid, 2 sets of chromosomes. Here we can see the longer blue and red came from mom, the shorter blue, or came from mom and dad, and the shorter blue and red one came from each parent. So this one has a total of 4 chromosomes. And then at the end, we can see there's 4 cells with only 2 chromosomes. So these are considered haploid. So again, where does meiosis occur in the body? The goal of meiosis, we said, is to make gametes. It's to make sperm and egg. So in what parts of the human body does meiosis occur? Where are sperm made in the human body? Okay, the reproductive organs, the testes in males, and eggs are made in the ovaries of females. So those are the only 2 places in the human body where meiosis is occurring. Everywhere else, all the growth, replacement, right, all that and repair is by mitosis. And those are happening in body cells. And the term we use for body cells is somatic. S-O-M-A-T-I-C. Somatic cells are body cells. And what did we say sex cells are called? It was on the first slide. The names of the cells. Gametes, yeah. So gametes are the names of the sex cells, and examples of gametes are sperm and egg. So their process of being formed is by meiosis, and somatic cells divide and repair and replace by mitosis. So body cells, mitosis, gametes, sex cells, meiosis. So the goal is we don't want to have carbon copies of ourselves in our kids. You don't want, a girl to be exactly half the mom, half the dad. That's not how it works. Have you noticed that when you look at people, they look like totally like their mother, or totally like their dad, or not like either parent, right? And that is because of the genetic diversity that happens in specific phases here within meiosis. So, key thing, make sure you highlight this. Meiosis brings about genetic variation. And a couple of things contributes to that genetic variation. And one of them is called crossing over. So here's some different chromosomes here. So these are chromosomes that when they cross over, which means they take the arms in prophase, the arms overlap to homologous chromosomes come together, and then their arms overlap and they exchange bits of chromosome. So here I can see this chromosome in the front here, not the ones behind. Let's just look at these in front. So let's say this is mom in red. So she's got A, big B, big C, big D on one, and then dad has all recessive alleles on his chromosome. Then they come together in prophase. The coming together we call synapsis, and we're going to get into this. You don't have to write it down right now. The coming together is called synapsis, and then they swap pieces, which is called crossing over, and this is what we have after that's done. Remember, this was all capital letters. Now it not inherited, but obtained a little piece of dad's chromosome on here. So now it's little c. And then if we look at the other one here, this one inherited a little bit of mom. So these two exchanged pieces here. So these chromosomes are no longer the same. So now we throw those into the phases of meiosis, and they line up on the middle, and they pull apart, and they pinch off, just like we saw in mitosis. But there's a lot of diversity now, because when these separate, we're not going to have the same information going into each cell, because the chromosomes started off as being a mixture already. So this happens in prophase, this coming together. So you could even write that here, prophase. So this happens in prophase, the crossing over. And then independent assortment happens in metaphase, and that just means these chromosomes line up on the metaphase plate, and they line up randomly, which means these chromosomes, when they line up, any four of these can line up on either side. So we could have two of the red ones going one way, two of the blue ones going one way, or I could have this one that's mostly dad and this one that's mostly mom. That can go into one cell. So there's a whole variety in how they line up in the middle. That's called independent assortment. Independent means random. Another name for this process is called random assortment. So let's go to the animation now, and we'll look at a couple of these things. I'm gonna do something here first. I'm just going to turn that animation off. Okay. So here we have a cell in prophase, and we recognize that as prophase, right? You've got, what do we call this loosely held DNA? What is it called? Chromatin. And then this nucleus, nuclear membrane, right? So in prophase 1, what would we expect to see? Chromosomes showing up, right? So in this case, we have 4 chromosomes. 2 look pretty similar, don't they? These 2, and then these 2 look pretty similar. So that means the green ones, let's just say, came from dad, and the purple ones came from mom. So we'll just assume that, okay? So mom's got a large and a small, or large and a small, and dad's got a large and a small chromosome. Now remember, if this was a human cell, there'd be 46 chromosomes in here, right? So let's say this is an individual, let's say this is a male making sperm. So it's important to kind of keep straight what's going on here. So these chromosomes from this male who's making sperm in the testes, these chromosomes came from his parents, right? So his cell, this cell that's going to undergo meiosis to make 4 sperm, are actually from a mixture of his own parents. So this is like grandma's, right? For what the sperm's going to use to make a baby, right? So the sperm contains grandma and grandpa's chromosomes, technically, right? So let's see what happens here. One of the first things that happens in prophase 1 of meiosis is the homologous chromosomes come together. We call that process synapsis. So what I would recommend is on a piece of paper, let's start with large circles. Let's use a piece of scratch paper, and we'll call this prophase 1. And then draw what's going on here. We've got this 4 structured thing. the name of this four structured thing after synapsis has occurred and we see these bundles of four sister chromatids, right? We call this whole structure here, we call that a tetrad. This one here would be a tetrad. And it's formed by a process. The coming together we call synapsis. So the tetrad was formed by synapsis. And that's prophase 1. So I can identify it in a diagram by just looking at those homologous chromosomes coming together to form that structure. Then, if we zoom in on a tetrad, we can see that they swapped bits and pieces of each other. What do we call that when they swap pieces? Crossing over. So crossing over occurs in prophase 1. So you can just make a little note underneath your diagram of what you see in this part of the animation here. Just put a little note, crossing over occurs. So they swap those arms. So now we've got some diversity. So they're not going to be the same. Then, just like we saw in mitosis, the centrioles move, they secrete that spindle, it attaches to the kinetochores of the centromere and pushes them to the center. So we call that metaphase 1. So in metaphase 1, what happens by the end of metaphase 1, we have these four structured things in the middle of the cell. But again, we call it random assortment or independent assortment because this one, this purple one on top here, could have been here, right? And this purple one here could have been here. So when this cell divides, we could see a variety of chromosomes depending on which one lined up where. So again, this is called independent assortment. So if I asked you on a test, where does independent assortment occur? I'm going to make this easier. Just go like this. Make these as X's. It's a lot easier. So independent assortment happened here.
What is that again?
That's when they can line up on the metaphase plate anyway, which means this green, mostly green one, this mostly purple one, it could have flipped over and lined up the other way. Because if we're looking at north-south, this green one, when this spindle fiber shortens, this green one's going to go north, right? This green one here is going to go north, and this purple one's going to go south. And this purple one's going to go north, and this one's going to go south, right? Well, what if I flip these two over? The green one would go that way, and the purple one would go that way. So this is going to eventually divide and become two new cells. So if I flip these over, like right now, this one's mostly mom is going to this cell, and this one's mostly dad, so whatever traits are on these are going to be a real mixture. But if these were flipped around, this side of the cell would get mostly mom's information. Wouldn't you agree? So that's what I'm saying. It doesn't matter which side they line up as long as they line up in the middle. And that's independent assortment. So then we continue, and just like in mitosis, the spindle shortens. That's anaphase 1. And there's nothing really to report going on there. So anaphase 1, we're just separating the tetrads, right? Now we've got this. Right? So we're separating the tetrads. So we're in anaphase 1, and we know we're in anaphase 1 because we're dealing with these X-shaped structures the whole way through. Then, just like we saw in mitosis, cytokinesis is going to happen. The chromosome is going to reappear. Not the chromosome, the nuclear membrane is going to reappear. And we have two new cells. But we're not done because meiosis 1 is only the first half of meiosis. Meiosis 2 is next. So we've got telophase 1 now, like this. We've got our chromosomes here and here, formed a nucleus again. But the big difference now going into meiosis 2 is that we're not going to replicate DNA in interphase. like we did in Meiosis 1. Because remember, Meiosis 1 started with the X's. Those were because of replication and interphase. So when we pinch this off, here's my 2 cells. Now each one of these cells is going to go through Meiosis 2. So this cell is going to divide, and this cell is now going to divide. And it's just by simple mitosis. Nothing fancy is going on here. So each of these is going to divide in Meiosis 2, which is just like mitosis. So, and don't we start out with X's in mitosis? Right? We replicated the DNA, and so now what's going to happen? They're just going to be scattered around the cell, and they're going to line up in the middle, just like we saw in mitosis, which was what your test was on. And then we're going to pull those sister chromatids apart in anaphase. But now remember, we have 2 cells that we started with. They're doing this simultaneously. Then at the end of anaphase 2, they're going to split. And how many chromosomes do we have now? Or how many cells? I should start with. How many cells? 4 cells. So each of these is going to split. And how many chromosomes did we start with in this example? Do you remember? Let's go back. We started with 4. At the end of Meiosis 1, we only had 2. So at the end of Meiosis 2, all we're doing is splitting those sister chromatids just like we do in mitosis. So we still have 2. So the reduction in chromosome number happened at the end of which phase? When did we go from diploid to haploid? When did we go from 4 chromosomes to 2 chromosomes?
Phase 1.
Yeah, the end of telophase 1, the end of Meiosis 1. So we call the Meiosis 1, we call it the reduction division because we took the number of chromosomes starting out as diploid and separated and pulled them apart to make them haploid. And again, Meiosis 2 is just like, Meiosis 2 is just like mitosis, that we just separate the sister chromatids. Now, what do we have at the end here? We have 4 cells, and these would be 4 sperm cells if we were doing this in the testes, or this would be 1 egg and 3 polar bodies if we're in the ovaries. Okay, so continuing our diagram then. Okay, so the key thing is meiosis is occurring in the ovaries and testes, but in males, this process is ongoing. They're constantly making sperm throughout the life. It slows down as men age, but it occurs throughout life, and it makes 4 cells. It's a continuous process, so there's no nuclear membrane formation or anything between Meiosis 1 and Meiosis 2. It just continues till we have 4 sperm cells. In the eggs, this process of meiosis actually occurred before baby was born. In utero, when a girl fetus is developing the ovaries, this process of meiosis is occurring during fetal development. So there's millions of eggs that are present in the female before birth, and then over time, up until puberty, they start to atrophy, and there's only a set amount that are actually ovulated. About 300 over a woman's lifetime are available for ovulation, and ovulation happens once a month when that egg is released. So all the eggs in the adult female are stuck in metaphase 2. They don't divide or do anything yet. They're just stuck in metaphase 2. And then when a sperm enters an egg at fertilization, conception, then that egg quickly completes meiosis 2, and then the process of development of the zygote occurs. So this is why when a woman has old eggs, when she's 40 to 50, early 40s, people can still conceive, but beyond that, it's pretty, much less common, very rare actually. So you can see whether it be chromosomal problems, because if that spindle is brittle in old eggs, and now we have to split these chromosomes apart after fertilization when sperm penetrates the nucleus, sometimes it'll break and leave a chromosome behind in the other cell. And that's where you end up with Down syndrome, where you have, why is there 3 chromosome 21s? Well, that's because those chromosomes, those sister chromatids should have split, but they didn't because the spindle broke and it left 2 chromosomes on one side of the cell. So you got the dad's sperm carrying 1 chromosome 21 and then 2 of the mothers that are left there because they didn't separate properly in anaphase. And that's where you see chromosomal issues. Okay, so let's go back to our notes. So we talked about independent assortment. Fertilization is the term we use, or conception is another word. It's the union of male and female gametes. So when the nucleus of the sperm combines with the nucleus of the egg, we get 46 chromosomes again. So if you think about, there's 23 chromosomes, and we have 2 of them. There's this many different chromosomally different zygotes possible. So it's amazing when you look at this huge number that we can have brothers and sisters that look so much alike, because there is a lot of genetic variation. So we talked about asexual reproduction in bacteria. What is that called? Binary fission. So that's genetically identical. And then we talked about cloning, right? Reproductive cloning produces genetically identical individuals. But when we have sperm and egg from 2 different individuals, and each of those sperm and egg were formed by meiosis, which took that parent, those parents, the grandparents' information, and scrambled them up to make sperm and egg, we see a lot of variation. So when we look at genetic variation, that's good when there is a lot of environmental changes, right? When we need to adapt to our environment. I think somebody mentioned that in the last class. You know, it's good for evolution for us to have genetic variation, right? If we have asexual reproduction, binary fission, that's good if this environment is stable. So that's why bacteria can do okay, right? Because they get into a human tissue and they just divide until we give them antibiotics, get the individual antibiotics, and that kills it. So how does antibiotic resistance occur then, do you think?
What was the question?
How does antibiotic resistance occur in bacteria if they're undergoing rapid binary fission? Yes, mutation. So if a mutation occurs that allows them to survive, who's going to survive? Only the ones that have that mutation. So that wipes out the other bacteria, right? They're killed by the antibiotic, and the ones that undergo mutation survive. And which ones undergo mutation eventually, do you think? The first ones, you know, when someone's first sick and takes their antibiotic, or when they leave the pill bottle at home next to their bed and they're off at work and school because they're feeling better? Those last bacteria, right, that survived up to that point, those are the ones that are more likely to develop a mutation to be resistant. So that's why it's so important when you're prescribed antibiotics that you take it for the full 5 to 7 days or till gone, right? Read your prescription bottles. And especially if you end up with like strep throat or recurrent sinus infections or recurrent urinary tract infections, it's really important to take those antibiotics all the way to the end. Because if you don't, the chances of mutation and resistance to that bacteria is going to go up. And we're going to look at that a little bit next week when we look at bacteria in the lab. So asexual reproduction is okay when the environment is stable and unchanging. But when we want variety is when there's changes in the environment. We can get some type of advantage and evolve. So, prophase 1. Spindle forms, nuclear envelope disappears, sister chromatids are present. Remember, the homologous pairs line up side by side. We call that synapsis. Make sure you highlight or underline that word, synapsis. And then when we have those homologs next to each other, the term for those 4-stranded structures are called bivalents. Another term is a tetrad. So either way you want to call them is acceptable. Tetrad, I think, makes more sense because there are 4 chromatids together. So then again, what's unique about Metaphase 2 is that the tetrads line up independently of each other. That's that independent assortment. Anaphase 1, the chromosomes separate. So just the tetrads separate. So you have two X's that are bound together. Now the X's separate from each other. The X's themselves don't separate, just the X's move, and you can see that in the diagrams you wrote. And remember, DNA, so interkinesis is the brief period between Meiosis 1 and Meiosis 2. It's not called interphase, it's just called interkinesis. Because it's a very short resting state. Then prophase 2, we still see chromosomes in metaphase 2 chromosomes lining up. This is just like mitosis. No different process occurs in meiosis 2 than we saw in mitosis. So as long as you know what mitosis is, that's meiosis 2. And then we end up with 4 haploid cells, all genetically unique, which means different, right? So this is from your textbook, just showing this process. I guess we do see nuclear membrane reforming here. I've never paid attention to that before. So it briefly reforms and then breaks down again in interphase. I'm not going to hold you accountable for that, so don't worry about it. It's a minor detail. But the key thing is where this is occurring is in the ovaries and testes, and what is the goal? Genetically variable haploid gametes. That's what we're making when we talk about meiosis and the function of meiosis. And this occurs in animals and plants. When I was over at UWL teaching labs there, we looked at ferns, and ferns actually have swimming viable sperm, just like we see in animals. So this occurs in plant cells and animal cells for variety and diversity. So if we compare the two side by side, meiosis has 2 nuclear divisions, right? Meiosis 1 and meiosis 2. Chromosomes synapse and cross over in prophase 1. We don't see that in mitosis. Centromeres are still present in meiosis 1 because those tetrads that formed are the only things that are separated in meiosis 1. Where in mitosis, we separate sister chromatids. We break apart those X's. The chromosome number goes from 2n to n in meiosis. It stays the same, 2n to 2n in mitosis. And they're identically or genetically different, and it's only for sexual reproduction. These are the same, and it's for growth or asexual reproduction like binary fission. So that's just a... So now what I have for you is a little homework assignment. It's 3 pages using the terms that we just talked about. And there'll be a short, this is just a very short little unit on meiosis, so there'll be a short quiz on Monday, just about, I don't know, 10 to 12 questions about meiosis. We're not going to have a formal sit-down lecture quiz. So study your notes here, study this worksheet, and you've got to go on. The rest of the time to get started on that if you like, or you can take it along, and I will see you on Monday.
I know you said this before, but